welcome to PW English YouTube channel. I'm Shalini Somashekhar, your botany teacher. I welcome you to today's lecture in the UMEED NEET English batch. In this video, we are going to be discussing the chapter Anatomy of Flowering Plants. In our previous lecture, we spoke in detail about the morphology of flowering plants. I hope all of you watched that lecture and understood everything very well. So, as always, in this batch, we are going to cover each chapter in detail in one shot. That is what we'll do today as well. Before we start off with the lecture, let's quickly go through how many questions have been asked from this chapter over the last few years in the NEET examination. Right here is the data from 2016 to 2024. You can see how roughly year after year, around 2 to 4 questions have constantly been asked from this chapter. So that's just how much you can expect from this chapter in 2025 as well. Last year in 2024, after the syllabus reduction, 2 questions have been asked. Now, another question that most of you all will have is whether to refer the old NCERT textbook or the new one for this chapter. For this chapter, I would suggest you to refer the old NCERT textbook. Okay, in old NCERT, you have tissues, different types of plant tissues. Then you have something called tissue systems. Then anatomy of different plant parts, where you take sections of dicot and monocot, root, stem, leaves and all of that. And then in the end, you have something called secondary growth. Among these concepts, secondary growth is not part of the NEET syllabus. So this you can omit. You don't have to study that. The remaining 3 parts or 3 concepts, tissues, tissue systems and anatomy, this is part of your NEET syllabus. So this has to be studied from old NCERT. In the new NCERT, even this is not there. This is deleted and it starts with this. But last year we saw how questions from tissues were asked. That is part of the syllabus. So you have to study everything in the old NCERT textbook except for secondary growth. Okay, so let's get started. First of all, what is anatomy? This chapter is called Anatomy of Flowering Plants. What does anatomy mean? Anatomy is a branch of biology where we learn about the internal structure of different organs or different parts of an organism. Over here, we are learning about anatomy of specifically flowering plants. What are flowering plants? Flowering plants are a group of plants which we also call angiosperms. In the previous chapter, we learned about the external features or morphology of angiosperms. Here, we are going to learn about anatomy of angiosperms. If we have to learn anatomy, if you have to learn about what kind of tissues are present within a plant organ, it could be root, stem or leaf, it's imperative that we take sections of that particular plant part, right? If you have to look inside the plant and see what kind of tissues you find, how are they arranged and all of that, it's important that we it's important that we take sections of different plant parts, look at it under the microscope, observe and find out what kind of tissues are present, right? That's exactly what we do in Anatomy of Flowering Plants. That's exactly what we learn in Anatomy of Flowering Plants. So you can broadly divide this chapter into 2 parts. In the first part, we will learn about the different types of tissues that exist in plants. We're not learning about where it is present and all of that. What are the different kinds of tissues that you can find in plants? First, you will learn about what are the features of those types of tissues, what could be their possible functions and all of that. In the second part, we will specifically learn about which tissue is located. Like if you consider a root, which tissue is located where inside the root? How is their arrangement? How is it in the stem? How is it in the leaf? How is it in monocots? How is it in dicots? So exactly how their arrangement is within different plant organs, we will learn in the second part. So in the first part, let's start off with plant tissues. This is not something that you're learning for the first time. I'm sure you've learnt about all of this, even in your lower grades in high school, probably 9th grade, 10th grade. This is like a quick revision. Because it's repeated, it's deleted from your new NCERT, but it's still part of the syllabus. Questions can be asked from this, so we will cover it, okay? So what are tissues? Tissues are a group of cells, right? We know about that. We've learned the definitions multiple times in our lower grades as well. What is a tissue? Tissue is a group of cells. Can you call any group of cells as a tissue? No. For a group of cells to be called a tissue, it should have a common origin and it should perform a common function, similar to how you perform group projects in school, right? You have like 4 or 5 people doing the same project. It's very similar. You have many different cells, but all of them are involved in performing just one job. Then we call it a tissue. So how can we define a tissue? It is a group of cells which has a common origin. Origin means they would have formed from the same type of cell. Okay, so their origin will be the same, and also they will perform one common function. Then you can call it a tissue. Here we are going to learn about plant tissues. You can broadly classify plant tissues into 2 categories. Now this classification is based on one very simple criterion. The criterion is that you have to check whether the cells in the tissue have the ability to undergo cell division or not. When we spoke about morphology of flowering plants, remember I told you about the morphology of root. We studied about the different regions of a root. There is a root cap, there is region of meristematic activity, and then there is region of elongation, and then there is a region of maturation. We learnt about all of this. If you remember, I told you that in the region of meristematic activity, there are cells that have the ability to divide, which they eventually lose. They will increase in size, and then they will stop increasing in size and take up permanent functions and become mature, right? This idea we've learnt about it in our previous chapter. So here, the criterion for classification is whether the cells in a tissue, can they undergo cell division or they cannot. Very simple. If a type of tissue can undergo cell division, then we call it meristematic tissue. If it cannot undergo cell division, then we call it permanent tissue. Okay, so plant tissues are broadly categorized into 2 types, meristematic being the first type and permanent tissues is the second type. How does this classification, what is the basis for this classification? The basis is whether they are capable of cell division or not. If they are capable of cell division, if the tissue contains actively dividing cells, then we call it meristematic tissue. If the cells have lost their ability to divide, they can no longer divide, then you call it permanent tissues. Okay, so this is the broad classification of plant tissues. We will learn in detail about meristematic tissues, its types, permanent tissues, its types as we move on. Let's start off with meristematic tissues. Meristose, why do we call it meristematic? What does meristematic mean? Does that mean they're present in the stem because meristem? Not really. Meristem is a word that we've derived from Greek, the Greek language. In Greek, meristos means dividing. Since the cells in meristematic tissues divide all the time, they keep on dividing, we call it meristematic tissue. Now, in a plant, meristematic tissue is not present anywhere and everywhere. There are specific locations, specific regions where you can find meristematic tissue, and those regions are called meristems. Though in the term there is meristem, meristematic tissue can be found in other plant organs like roots also. Stem does not mean our plant stem here. Okay, so it is basically derived from the word meristose, that's why it's called meristem, but this does not necessarily have to be part of the stem. Okay, so meristems are those specific particular locations at which you can find meristematic tissue. We can classify meristematic tissue in many different ways. One way which we classify meristematic tissue is based on where it is located, based on its location. Okay, if you consider the location of meristematic tissue, you can divide it into 3 different categories. The first one is apical meristem. The second one is intercalary meristem. The third one is lateral meristem. Apical meristems are usually found at the apices of plants. Apical means what? Apex. Apex means what? Tip. Right? Tip is also called the apex. So apical meristem is generally found at the tip of the stem or the shoot and the tip of the root. Remember when we studied about the different regions of root in morphology of flowering plants, I told you there is a region called region of meristematic activity. Cells are actively dividing there. That region has to be protected. That is why there is a root cap and all of that, right? So it is present at the tip of the root. It's also present at the tip of the shoot. Talking about intercalary meristem. This you generally find in case of monocots. Very rarely you can see them in dicots as well. I told you meristems are the types of tissues that have the ability to divide. Permanent tissues have lost that ability to divide. In between these permanent tissues in certain regions, if you find meristematic tissue, then we call that intercalary meristem. Okay. Third is called lateral meristem. Lateral meristems are present on the sides. Lateral means what? Side. So if this is a stem, I'm taking a longitudinal section of the stem. If this is the stem, let's say, I'm cutting the stem like this. If this is the outermost layer of the stem, the epidermis, inside, on the sides, you can see lateral meristem. It's like, you can think of it like a pen and the refill that is present inside the pen. The outer body of the pen represents the outer body of the stem. Inside, similar to how you have a refill which is cylindrical, within the plant stem, you will have a cylinder of meristematic tissue, which we call the lateral meristem. Because if you cut it like that, you will see two lines, right? That is why on the sides. That's why you call it lateral meristem. But it's not like necessarily present only on the sides. When you take a section, it appears to be present on the sides, but it's actually a cylinder inside. Okay, more about each of these we learn in detail, starting with apical meristems. Apical meristem is of two types depending upon its location again. If it is present at the tip of the root, we call it root apical meristem. In short, it's represented as RAM. We've already learnt about it. This is an NCERT diagram. There is a root cap in roots, and then there is a region of meristematic activity that itself represents the root apical meristem. Then, if it is present at the apex of the shoot or the tip of the shoot, as you can see in this case, then we call it shoot apical meristem. See here, all of these cells that are present at the tip, that is the shoot apical meristem. Remember, it is from the shoot apical meristem that leaves will arise, leaf primordia forms, right? So, whatever you're looking at here, which I will outline in green color, are basically leaf primordia that are arising. Eventually, they will become big leaves. Okay, so this is the leaf. So if it is present at the tip of the shoot, then you call it the shoot apical meristem. Next, already you know, at the root tip and the shoot tip, apical meristems are present. We've already learned in our previous lecture that if you think of this as a stem, the leaves will arise at specific points on the stem, specific regions of the stem, which we call nodes. These are the nodes. And at the axil of the stem, the angle between the leaf and the stem, small buds will be present. These are called axillary buds. This is something that we've already learned, so I'm going quick, right? And these axillary buds could either form a branch, they could develop into a flower or whatever, okay? Now, in the axillary buds also, you have meristematic tissue. How did the meristematic tissue get to the buds here? We'll try to understand that. Let's take the shoot tip. If you look at the shoot tip very closely, of course, when you look at it with your naked eye, you will not be able to make out. But if you take like a section of the, if you cut the shoot tip and look at it under the microscope, you can see that at the tip, you will have meristematic tissue. From the sides of the shoot tip laterally, you can see small leaf-like structures or appendages present. We call them leaf primordia. Right now, you don't yet see them as leaves. They are not large enough to be seen as leaves. But what we can say is that they are going to become leaves in the future. How do they become leaves? What happens when they become leaves? this region between this leaf primordium and this leaf primordium, it will start elongating. It will start becoming longer. Okay, so that internodal region will start elongating, increasing in length. When that happens, you can see how, if this was your shoot apical meristem, these leaves are becoming bigger. You can see how some of these meristematic cells that were present at the tip will be left behind here. Whatever cells from the shoot apical meristem are left behind here as this grows and develops, that is what becomes in the future the axillary bud. In the axillary bud also, you can see that there are meristematic cells. So, apical meristems, what do? Where do you find them? They occupy the tips of roots and shoots. During the formation of leaves, some cells left behind from the shoot apical meristem form what we call this axillary bud. How did this axillary bud come about? It formed because of some of the meristematic cells from the shoot apical meristem that were left behind during the formation of leaves. Okay, so that is what forms the axillary bud. Next, let's talk about intercalary meristem. You don't have to know much about it. You mostly find this in grasses, does not mean it doesn't occur in dicots. Very rarely you can see this in vines and other types of plants in dicots as well. So mostly they occur in grasses. Inter means in between, intercalary, in between what are they present? They are present in between permanent or mature tissues. That's why you call it intercalary meristem. Generally you see these herbivorous organisms like cattle, cow, sheep, buffalo grazing on plants, right? If they feed on grasses, grass doesn't necessarily mean the grass that's growing on the soil like small grasses. Even wheat, jowar, maize, ragi, all of these, rice, all of these are included in the grass family itself. If a herbivorous organism comes and feeds on it, this intercalary meristem that is present will help in regenerating whatever parts the herbivorous organisms would have eaten up. Okay, so that is what its function is. Now let's talk about lateral meristem, which is the third type of meristem. This you can find only in dicotyledonous plants and in gymnosperms. If it is angiosperm, you can find it in dicots only. You don't find them in monocots. In angiosperms, you can find that only in dicots, and in gymnosperms also you can find lateral meristem being present. Okay. Now, where do you find this? This also forms in the mature regions of the plant parts, plant organs like roots and shoots. Like I've already mentioned here, they are cylindrical. Okay, like this, they are cylindrical. The reason we are calling it lateral is because if you cut it like this horizontally, they will appear to be present on the side, right? If you cut a refill like this, you will see two lines on the side, right? Very similarly, if you cut a pen like this, you will see two lines of the refill. Like that, you will find two sides, on the two sides, the tissue being present. That's why we call it lateral meristem, but actually inside the plant, they are cylindrical. Examples for this you must remember: fascicular vascular cambium and interfascicular cambium and cork cambium. Okay, there are three examples here, 1 and 2 and 3. Fascicular vascular cambium has another name. It is also called intrafascicular cambium. Okay, it's called intrafascicular cambium. So you have three types: intrafascicular cambium, interfascicular cambium, and cork cambium. If you think this is a dicot stem and I take a section, transverse section of the dicot stem, if I'm cutting it horizontally, this is horizontal, right? Yeah, this is vertical. Yeah. If you're cutting it transversely like this horizontally, take like an extremely thin slice, put a put it on the slide and look at it under the microscope, what you will see will appear in circular outline. The stem will appear circular because stems are generally cylindrical. If you cut it like this, it will appear circular, right? So in this, you can find, rings of cambium present towards the outside. Where exactly I'll explain later. For now, just understand. Towards the outside, near the cortical region. Why can't I find a center? Okay. You can find one ring of cambium, and slightly, relatively towards the inside, you can find another ring of cambium. Okay. So this cambium that is there inside, towards the inside, this will contain in some regions in some regions within the same ring, you will find intrafascicular cambium. Within the same ring, in certain regions, you find interfascicular cambium. Okay, and towards the outside, you will find cork cambium. Earlier, you had this concept called secondary growth, where we used to teach about how exactly intra forms, inter forms, cork cambium forms and all of that. Right now, it's deleted. You don't have to learn about the details of how these cambial rings form. Just know that they exist in the form of cylinders inside the plant. If you take a section, it will appear in the form of rings. Okay. So within inside you have one ring. This one ring that includes your intrafascicular and interfascicular, both of these together. It is called vascular cambium. And what forms towards the outside, that is called cork cambium. Okay, so these are present inside the stems in dicots. Okay, now that is about your meristematic tissue, lateral meristems. Okay, now I told you there's another way of classifying meristematic tissue. It is based on what kind of growth it contributes to, one thing. Secondly, when was this meristem formed? Okay, these two things are the criteria here. What are the two criteria here? What kind of growth does it contribute to? Does it cause an increase in the length of the plant, or does it cause an increase in the girth of the plant? This is one question we have to ask. And secondly, was it always formed? Was it always there from the beginning, or it formed later? When was it formed? Okay, you know, meristematic tissue contains actively dividing cells. One cell divides to give you two cells, 2 to 4, 4 to 8 and so on. So when the number of cells is increasing in a plant, then obviously that plant part where that meristem is will grow. If there is meristem present at the tip of the root, tip of the shoot, if there is active cell division happening there, as a result of that active cell division, the root tip will grow, the shoot tip will grow. So, right, because of its ability to undergo cell division, the meristematic tissues will contribute to growth in the plant. Now, growth can happen along two ways, in two ways. There can be an increase in the length of the plant, length of the root, length of the shoot. Also, there can be increase in girth of the stem and root. Girth means basically diameter. Like a small mango seed, if you plant it, it will become like a small sapling first and then eventually it grows. When a small mango sapling grows into a mango tree, what happens? It's increasing in length, it's increasing in height, it's growing taller. Simultaneously, the stem is also growing wider and becoming that big trunk that we see in a mango tree. So in dicot especially, you see two types of growth. Growth increase in height, increase in girth. If there is increase in length, if there is an increase in length that you see because of meristematic activity, such type of meristems which contribute to increase in length, they are called primary meristems. Secondary meristems are those that cause an increase in girth. Primary meristems have been present in the plant since the beginning, since the plant germinated and formed, since the beginning, primary meristems will be present. Secondary meristems form much later. Okay, so these secondary meristems are not there from the beginning. If they're not there from the beginning, how can they form later? Whatever permanent tissue will be there in the plant which has lost the ability to undergo cell division, if those permanent tissues regain the ability to undergo cell division and become meristematic, then we call it secondary meristem. Okay. If meristems have been there since the beginning, after germination, since the time the plant has germinated, the seed has germinated and a small plant has formed, since that time, if the meristem is there in the plant, we call it primary. If it develops later because some permanent tissues regained the ability to undergo cell division, permanent tissues would have lost that ability. If they regain that ability and become meristematic again, then such meristems are called secondary meristems. So for examples, primary meristems include your apical meristem as well as the intercalary meristem. Both of these contribute to increase in height. Now the secondary meristem, example for this is all your lateral meristems, all the cambial rings. This is your vascular as well as cork cambium. Let's understand this. If you look at this, apical meristem is here. If there is cell division and more cells are forming, there will be increase in length. You have intercalary meristem here. If these are dividing and forms more cells, then there will be increase in length. There are lateral meristem or cambium present here. How these cambial cells divide is, I will tell you, they will divide in this plane. If you had one cell layer, imagine that cell layer is giving you dividing in this plane such that from one cell you have two cells. From two cells you have four cells. Every time cell division is happening periclinally in the same plane. Again, this cell divides, it will add more cells to the inside and outside. So what happens when cells are dividing in this plane? More cells are added towards the inside and outside. As a result, there will be an increase in the girth of that particular plant part. For example, stem. So if inside the stem there is a layer of cells which has the ability to divide, and it is dividing cells always in this plane such that more cells are added towards the inside or outside of this ring, that will contribute to increase in the girth. Okay, so this vascular cambium that includes interfascicular, intrafascicular and the cork cambium, in that cambial rings always cells will divide in the periclinal plane itself and cause an increase in the girth of the stem. Okay, so that's about your meristematic tissue. Now let's talk about permanent tissues. What are permanent tissues? I've already told you. Permanent tissues are simply those tissues that have lost their ability to divide and taken up certain specific functions inside the plant. Then we say that the cells or the tissue is mature or it is differentiated, or we can call it permanent tissues. Okay. So the cells that are formed by the meristems that become structurally and functionally specialized and lose their ability to divide, that is called a permanent tissue. All of these permanent tissues, they arise from meristematic tissue itself. Meristematic tissues have only one job to perform. They always keep on dividing, add more cells to the plant and contribute to its growth. Apart from that, it doesn't do much. But in a plant, there are so many functions to perform, right? The plant has to be protected. In plant, conduction of water and minerals have to happen. In the leaves, photosynthesis has to happen. Photosynthesis has to happen. So if all of these functions need to happen, then if the cell is keeping on dividing and not really doing much, it won't help if you have just meristematic tissue. So at one point, one subset of the meristematic tissues, if you have like 100 meristematic cells, hypothetically, then one subset of it, probably 40 or 50 of cells, stop dividing. While the remaining 50 continue to divide and give rise to more cells, one subset of it will stop dividing. What do they do then? They will take up specific permanent functions in a plant. For example, they would want to provide mechanical support. In order to provide mechanical support, whatever modifications they have to undergo, they will undergo all of those changes and modifications, and they will become fit to provide mechanical support for the plant. If it has to perform photosynthesis, then the meristematic cell will stop dividing. It will, undergo whatever modifications are required for photosynthesis. It will develop more chloroplasts, it will form more chloroplasts inside, and it will become fit for photosynthesis. If it has to, take part in transport of food and water, then it will, modify itself accordingly and help in the process of transport of food and water. So according to its function, it will modify itself. Okay, similar to how we have different career options. Like if I have to become a good teacher, then I have to modify myself accordingly. Like when I finished graduation, I had so many options in front of me. Meristematic tissues also have a lot of options in front of them. They could become any different type of permanent tissue. So I had a lot of options. I could get into research, I could start teaching, I could probably, become an artist by painting, I could probably pursue a career in music. So these are the different options. I decide that I want to be a teacher. I will acquire the skills that a teacher requires. I have to start writing more neatly on the board so that the students can understand. I have to speak at a pace which is very slow so that everybody can follow. I have to have good knowledge in the concept, so I have to study a lot. So these are the skills that I should, acquire in order to become a good teacher. Like that, meristematic tissues, when they are becoming permanent, they will have many options. They could become parenchyma, they could become sclerenchyma, they could become collenchyma, they could become conducting tissues like xylem and phloem. Depending upon what it becomes, what it chooses to become, it will modify it accordingly. Like parenchyma in, leaf performs photosynthesis. Xylem in leaf is involved in transport of water. So if it has to become parenchyma, it will modify itself accordingly. If it wants to become xylem, it will modify itself accordingly. So that process by which cells modify themselves and take up permanent functions and become modified accordingly, that process is called differentiation. And such tissues which don't have the ability, they don't care about cell division anymore, they have one specific function to perform, they continue to do that for the most of your life, for the most of their life, for the most part, then you call it permanent tissues. Okay, so now generally, these permanent tissues do not divide any further, but in rare cases, like I mentioned here earlier, if this cambial ring has to form, this core cambium has to form, that forms from the permanent tissue itself. Under such conditions, permanent tissues will give rise to meristematic tissues, but otherwise, in majority of the cases, you do not find permanent tissues gaining the ability to divide again. Okay, so in most cases they will stop dividing. Very rarely they can become meristematic again. Okay, now permanent tissues can be classified further. For the classification of permanent tissues, we classify them into two different categories broadly. The criterion that we use for classification here is whether the permanent tissue is made up of just one type of cell or it's made up of more than one type of cell. So how many types of cells are present in a tissue? I told you that a tissue is a group of cells, so more than one cell obviously will be present. Are all of the cells in a tissue of the same type, or are there different types of cells forming a tissue? That is what differentiates these two types of permanent tissues. If they are all made up of a single type of cell, all of the cells are similar, then you call it simple permanent tissues. If you have more than one type of tissue, more than one type of cell forming the tissue, then you call it complex permanent tissue. In simple permanent tissue, we have three types: parenchyma, collenchyma, sclerenchyma. And in complex permanent tissue, we have two types: xylem and phloem. First, we will learn about simple permanent tissues in this exact same order: parenchyma, collenchyma, sclerenchyma. And then after that, we will learn about xylem and phloem. Alright. Now, let's start off with parenchyma. Parenchyma is the most abundant type of tissue that you can find in plants. It is present in the root, it is present in the stem, it is present in the leaf, it's present pretty much everywhere inside the plant. It is the most abundant. This is important, you have to remember this. Okay, so it is the most abundant. Of course, if it's abundant, it will definitely form a major component within organs, right? Talking about the shape is variable. Generally, they are isodiametric, which means they are somewhat spherical in nature. But in different plant parts, their shape could change. They could be cylindrical in some plant parts. They could be oval, polygonal, elongated. So its shape can change. But in general, in most cases, they are somewhat isodiametric. Isodiametric means almost spherical. Like where you take the diameter, it will be similar. Nearly equal, then you call it isodiametric. Okay, so how are the walls here? You know, all plant cells have cell walls. Cell walls are generally made up of cellulose and pectin, but in different types of tissues, along with the cellulosic cell wall, there may be additional secondary walls that are made up of other substances. In parenchyma, you don't see such secondary wall thickenings much. The primary wall itself will consist of cellulose and it will be thin compared to the other cell types. In parenchyma, you can find thin cell walls. In collenchyma, sclerenchyma, they are relatively thick. These parenchyma cells are generally closely packed. Though they are closely packed, as you can see in this illustration here, this is what you have in NCERT. If you look at it, though they are very closely packed, not much space is left between each other. There will be small spaces that are present between the cells. We call them intercellular spaces. This is the diagram of NCERT. This is actually how parenchyma looks under the microscope. You can see even over here, there are small, not like too much of a space, but small spaces present between neighboring cells. We call them intercellular spaces. What is the function? Depending upon where it is present, its function can vary. If it's present in the leaf, it could possibly help in photosynthesis. If it's present as the outermost layer in any part of the plant like root, stem or leaf, it will obviously offer protection. If it's present in a storage root, it could be storing reserve food. So depending upon where it is located, its function can vary. So photosynthesis, storage, secretion, all of these are different functions that parenchyma performs in different parts of the plant. Now let's talk about collenchyma. Collenchyma is found only in dicots and it's found in dicot stems specifically, right under the epidermis, right under the outermost layer. You don't yet know what epidermis is. Epidermis is the outermost layer in the plant basically. Root, stem or leaf, outermost layer is called the epidermis. It's just like how we have skin. The skin of plants, outermost layer is the epidermis. So right below the epidermis in dicot stems, you can find this. This can be found as a continuous homogeneous layer, or in some cases it can be found in the form of patches as well. This collenchyma is a living type of tissue and it offers mechanical support. Here, like all plant cells, in the cell wall, you can find cellulose. But along with cellulose, at the corners, you can see how there is secondary cell wall thickening. Now, this thickening is made up of generally pectin. Okay, what is the shape? It is oval, spherical or polygonal. They generally contain chloroplasts. Unlike parenchyma, over here, intercellular spaces are mostly absent. Its function is to provide mechanical support. They offer like some little flexibility and also they provide mechanical support. They are present in young stems like I told you earlier and also in the petiole. What is petiole now? We've learnt about it in the last chapter. Petiole is the stalk of the leaf. So they can be present there. Okay, that's all you need to know about collenchyma. Know these diagrams because diagram based questions can be asked. Last type of simple permanent tissue is sclerenchyma. Now this sclerenchyma is a dead type of tissue. If it is dead, what is its function? Why is it even there? You know, plants have to stand tall, right? And then it needs mechanical support. They don't have bones like us. Instead, they will have these sclerenchyma tissues which are dead. Dead meaning there is no cytoplasm inside, there is no metabolic activity that happens. While they form, I told you, when meristematic tissue becomes like a permanent tissue, it will have to undergo some changes. While a meristematic tissue undergoes changes to become sclerenchyma, it will lose its protoplasm and it will develop like really thick, rigid walls. The walls are so thick almost that the inside lumen inside the cell is completely almost obliterated. So it will develop very thick walls, and in the wall of these sclerenchyma cells you can find another substance called lignin, and that will provide a lot of mechanical support for the plant and make sure that the plant stays upright. If a plant is made up of only like soft tissues, then the plant cannot stand upright, right? It requires some mechanical support to stand upright, and that mechanical support is provided by sclerenchyma. Though they are dead cells, they have sacrificed themselves, they have like killed or they have died in order to provide mechanical support for the plant. So they are like martyrs. Yeah. So this sclerenchyma has thick lignified walls. Because they have such thick walls, inside whatever space will be present inside the cells, the lumen, that is almost completely covered. So you can see here, this is almost spherical. This is a type of sclerenchyma called sclerite. You can see this is the lumen. From here till here, it is the wall itself. Okay, so yeah, as you can see here, the outer wall, which is represented by red color, which I will represent by red color here, is almost smooth. This is the primary wall. Inside that you have the secondary wall, which here is represented by pink color. You can see how it's not uniformly present throughout. There are a lot of white spaces here where you don't find lignin, where you don't find the secondary wall thickenings. Such regions are called pits. Okay, so it will have a few or many pits present. Usually they are dead without cytoplasm or protoplasm at maturity. Their function is to provide mechanical support. You have two types of sclerenchyma. One is fiber, the one to the left, and the other one is sclerites. Fibers are generally elongated with tapering ends, and sclerites come in different shapes. Okay, so first let's talk about fibers. Fibers, regardless of whether it's fiber or sclerite, sclerenchyma is thick walled. Okay, so here talking about the shape, they are elongated and they have tapering pointed ends. They generally occur in groups. They can be found in various parts of the plant. Sclerites are also thick walled. They have a very narrow lumen because the cell wall obliterates the lumen. Its shape right here is spherical, but in different plants it can exist in different shapes. It could be spherical, oval, cylindrical. Where do you find them? You know, when you eat fruits like pear and apple, you feel like a gritty nature. That is because of the sclerites present in it. It's located in the fruit walls of nuts, pulp of guava, pear, sapota, leguminous plants like peas, beans. In the seed coat you can find them. In the leaves of tea you can find them. So these are the different locations of sclerites. That's about our sclerenchyma tissue. Now we are done with simple permanent tissues. What do we have next? Complex permanent tissues. What are complex permanent tissues? They are permanent tissues that have lost the ability to divide. They are called complex because they are made up of more than one type of cell. Under complex permanent tissue, we have two types, xylem and phloem. Both of them are conducting tissues. Xylem helps in the conduction of water and minerals. Phloem helps in the conduction of food. Okay? Yeah. Now, first let's learn about xylem and then we'll learn about phloem. What is xylem? It is one of the conducting tissues. I already told you. What does it conduct? It conducts water and minerals from the roots to the leaves. Apart from conducting water and minerals, xylem has another function which we generally overlook. It is providing mechanical support. In xylem, you have four different types of cells. I told you, if it is a complex permanent tissue, it will have more than one type of cell. In xylem, you have four different types of cells. Of the four different types of cells, tracheids, vessels and xylem fibers, these are dead. If they are dead, that means they can provide mechanical support. They are not soft, they will have thick walls, they can provide mechanical support. Xylem parenchyma is the only living type of cell that you can find in xylem. Okay, we learn about each of these in detail. Starting with tracheids. Tracheids are, you know, if water conducting tissue, if a conducting tissue has to conduct water, there has to be some space present inside. It has to be empty. If it's filled with cytoplasm and organelles, conduction of water has to happen against gravity. Remember, from the roots to the leaves, water has to be conducted. There will be a lot of resistance if living cells have to conduct water. So in order to make conduction of water easier process, the cells will become empty. They will lose their protoplast, they will kill themselves, they will sacrifice themselves and become xylem tracheids. And these xylem tracheids are empty inside. Through these empty cells, like this is one tracheid, right on top of it another one will join, one on top of each other. It will form like a long column. Empty cells, there's space inside. Through this column, if water gets transported, there is lesser resistance. As it is, there is a lot of trouble for water to be transported because it has to happen against gravity. If there is any resistance, then it will make it even difficult. So the plants very cleverly over the course of evolution have developed xylem and phloem, which are cells that have lost the protoplast and formed conducting elements. So in angiosperms, we are talking about angiosperms in this chapter. In angiosperms, tracheids are there, but not much water gets transported through the tracheids. But in gymnosperms, which also has xylem and phloem, in pteridophytes, which also has xylem and phloem, in xylem, tracheids are the major conducting element. Okay. Yeah, so these are elongated tube-like cells. These two are the tracheids. They are very narrow. You can think of them similar to be like Sclerenchyma because they also have very thick lignified walls. These tracheids have tapering ends. They look very similar to fibers, right? They are dead without protoplasm. Inner layers of the walls have thickenings which can vary, like pits I showed you, similar to that there will be differential thickening, and you can see that it varies in form. It could form like a reticulum, it could be spiral, they come in different types. It's not part of your syllabus, so don't worry about it. That's about tracheids. This is the major conducting element in gymnosperms, but in angiosperms probably less than 5% of conduction happens through this. Okay. In angiosperms, the major conducting element is the vessels, which is also called trachea. The reason angiosperms are called angiosperms is because angio means vessel, right? You've learnt about it in body fluids and circulation. Angio is about blood vessels. Similarly, here, the conducting elements in flowering plants or angiosperms is the vessel. That's the reason we call it angiosperms. So here, only in angiosperms, you can find vessels. These are vessels. This one is a vessel. You can see how they are relatively wider than the tracheids, right? They are cylindrical and wider. They are also made up of many cells. Individual cell is called a vessel member, and one on top of another it lines and forms a long column. Then you call it the vessel. Each cell is called a vessel member. They also, similar to Sclerenchyma, have lignified walls and a large central cavity. They are dead. They do not have any protoplast at maturity. This and its next cell is connected through perforations in their common walls. Perforations means pores. Okay. So remember this. This is a characteristic feature of angiosperms. You don't find this in most gymnosperms. Exceptions are there. You don't have to worry about it. Okay. So vessels are present only in angiosperms. If you look at the plant kingdom, bryophytes don't have vascular system. Pteridophytes, gymnosperms, angiosperms, these have xylem and phloem. But in pteridophytes and gymnosperms, you don't find vessels. Vessels are found only in angiosperms. Okay. Next, xylem fibers. These provide mechanical support. These are nothing but Sclerenchyma fibers. Alright. And then xylem parenchyma. This is the only living tissue you find there. Like all parenchyma, they have a thin wall. Their cell wall is made up of just cellulose. No much thickenings are present. They can store food materials in different forms like starch, tannins, etc. So that's about xylem. Here's what it looks like in the tissue. So here you see this is a tracheid. This is a xylem fiber here. This is the tracheid. Okay. And then, yeah. This is the tracheid. Here you have the xylem fiber. Here you have the vessel. And here you have the parenchyma. They are present together and perform the function of water transport, mineral transport. So that's why they have a common function. They are different types of cells. That's why you call it complex permanent tissue. That's about xylem. Now let's move on and learn about, before we move on to phloem, let me tell you about protoxylem and metaxylem. Xylem is the conducting tissue that forms inside, water conducting tissue that forms inside the plants, in the root, stem, leaves. When the xylem first forms, initially when the plant is still young, the vessels that you see, if you take this as a plant stem, xylem vessels are like a tube. So you can think of that refill like structure present inside the plant, right? So if you look at it, if you take a section transversely and look at it under the microscope, all of those vessels, since they are cylindrical, they will appear circular in outline when you take a section. So if you look at the xylem elements and the xylem vessels that are formed initially, their diameter will be small. The later formed xylem elements will have a wider diameter or lumen. Okay, so depending upon whether it is its lumen is smaller or larger, whether it was first formed or later formed, you can categorize it into protoxylem and metaxylem. Protoxylem is first formed. Here you can see protoxylem elements. These are smaller in diameter. Metaxylem is later formed and it is larger in diameter. In different plant parts, depending upon where protoxylem is, where metaxylem is, you have different types of arrangement, exarch, endarch. We'll learn about it. Okay? Yeah. Let's move on to learn about phloem now. Okay, wait, let me teach you this already. So, within the root or stem, you can find different types of arrangement based on the position of protoxylem. Protoxylem has a smaller lumen. So in this case, you can see that the protoxylem is present towards the center and the metaxylem is present towards the periphery. The larger xylem vessels are present towards the periphery. If protoxylem is facing the center, then we call such a type of xylem arrangement as endarch. Endo means inside. Overall, we are considering here the position of protoxylem. If protoxylem is towards the center, which we call the pith, more about this we'll learn later. If it's present towards the center, you call it endarch. If it is present away from the center towards the periphery, then we call it exarch. Exo means outside. This classification depends on solely the position of protoxylem, which is the smaller xylem element, smaller xylem vessels that are first formed. This type of endarch arrangement of xylem can be seen in stems. Exarch arrangement can be found in roots. Okay? Yeah. Now let's move on and learn about phloem. Phloem also is a complex permanent tissue. It is made up of 4 different types of cells again. This is involved in the conduction of food from across different parts of the plant. Okay? Over here, you have 4 types, of which 3 types of cells are living and 1 is dead. We learn about each of these one by one, starting with sieve tube elements. What are the 4 types of cells? You have sieve tube elements, companion cells, phloem parenchyma and phloem fibers. Okay, so yeah, we'll talk about sieve tube elements first. Sieve tube elements are characteristic feature of angiosperms. They are found in angiosperms only. They are absent in gymnosperms. In gymnosperms, you have something else that is called sieve cells. You don't call them sieve tube elements, you call them sieve cells. These are the major conducting parts. Similar to how vessels conducted water, here they conduct food, but vessels were empty dead cells, but sieve tube elements are living cells. They are also cylindrical in shape, tube-like, and they are arranged longitudinally, which means they are arranged one on top of each other like this to form a continuous tube. If this is 1 cell, 2 cell, 3 cell, there are walls that exist between these cells, right? They are called cross walls. So the cross walls or the walls that are present between 2 cells, they will be perforated. If they are, completely present, if there is no perforation or pore present here, then how will food be transported from one cell to another? If it has to be transported, there should be some way it can pass from one cell to the other cell, right? So the cross walls here will have a lot of perforations, and such cross walls are called sieve plates. Because the sieve tube elements are involved in the conduction of food, it requires a lot of space. These sieve tube elements would have lost their nucleus during maturity. You know, we talk about nucleus a lot, we say nucleus is important, but these sieve tube elements would have lost their nucleus. Similar to how our RBCs lose the nucleus, even these sieve tube elements become enucleated when they mature. Okay, so that allows them space for transport of food. Yeah, now they have perforated cross walls or end walls. We call them sieve plates. I already told you about that. Since they lack a nucleus of their own, they are associated with cells that are called companion cells. The nucleus of the companion cells governs the activity of the sieve tube elements. This doesn't have a nucleus of its own. How can its functions be governed? The companion cells lying adjacent to the sieve tube cells, this will govern the activity of the sieve tube elements. Okay. In a mature sieve tube element, here I am going to highlight that, outline it in blue color. This is a sieve tube element, mature sieve tube element. Okay, this is one of them. You can see how in the cross wall or the end wall there are perforations, there are pores present. You will have a large vacuole in the center. Because there is a vacuole present, cytoplasm is present towards the periphery. Like I already mentioned, this does not have a nucleus. Though it does not have a nucleus, its metabolic functions are controlled by companion cells. Okay, now let's talk about companion cells. Again, these are absent in gymnosperms. They are found only in angiosperms. In gymnosperms, instead of companion cells, you have something called albuminous cells. Companion cells are basically special type of parenchyma itself. Like I mentioned earlier, they are very closely associated with sieve tube elements. There will be pit fields. If this is one sieve, wait, let me use a different color. This is one companion cell. This is one sieve tube element. This wall is common between this cell and this cell, right? In this common wall, there will be certain pit fields present that will help in the communication between these two types of cells. Also, for transport of food along the phloem or through the phloem, through these sieve tube elements, a gradient has to be maintained, and that apparently is maintained. The pressure gradient is maintained by the sieve tubes. The companion cells help the sieve tubes in maintaining a pressure gradient. Earlier you had a chapter called Transport and Plants, where we would learn in detail about how phloem transports food, how does xylem transport water, but right now in your syllabus that chapter is deleted, so you don't have to know much about how exactly they maintain a pressure gradient or anything. Okay, so that's about the companion cells. Then we have phloem parenchyma. Phloem parenchyma, important point, it's absent in most monocots. In the phloem, in monocots, parenchyma is generally absent. Yeah, so parenchyma here is somewhat elongated, tapering with cylindrical walls. They have a dense cytoplasm, prominent nucleus, cell wall is made up of cellulose, typical parenchyma characters. And then, just like how between two neighboring cells in plants you have plasmodesmatal connections or cytoplasmic connections, even over here you will have plasmodesmatal connections present. And its function in phloem, parenchyma's function in phloem is to store reserve food and other substances like resins, latex, tannins, mucilage stuff. Last one is phloem parenchyma. Phloem fiber. Phloem fiber has another name. It's also called bast fiber. If it's a fiber, then it's obvious it's sclerenchymatous. Again, similar to how you had primary xylem, secondary xylem, you have primary phloem and secondary phloem. Primary phloem is first formed phloem. Secondary phloem happens during secondary growth when there is increase in girth. Okay. So this phloem fibers are generally absent in primary phloem. Phloem parenchyma is generally absent in monocots. Phloem fibers is generally absent in primary phloem. You should remember this. Okay. and it is found in secondary phloem. Generally, you will be asked statement-based questions from this chapter, and there will almost always be like one statement with exceptions like this. Okay, so you should remember that in gymnosperms there is no vessels. In gymnosperm there is no sieve tube element. Instead they have sieve cells. Instead of companion cells they have albuminous cells. In monocots you do not find phloem parenchyma. In primary phloem you do not find phloem fibers. So these are things that you have to make note of and remember really well. Okay, yeah. Phloem fibers as always will be elongated if it's a Sclerenchyma fiber, and they will have pointed needle-like apices. They have tapering ends for sure. The cell wall always will be thick since this is Sclerenchyma. They will become dead at maturity. Commercially, phloem fiber is used. Jute, flax and hemp. These fibers are obtained from plants. They make jute saris, jute bags, jute pencil pouches, jute cases for mobile and all of that, right? So that is obtained from the phloem fiber itself. Yeah. Similar to how we saw protoxylem and protophlloem, you have something called protophlloem and, earlier we saw protoxylem and metaxylem. Even in phloem, you have protophlloem and metaphlloem. In protophlloem, sieve tube elements will be smaller in diameter. They are first formed. In metaphlloem, sieve tube elements will have a larger diameter. They are later formed. Very similar to protoxylem and metaxylem. Okay? Yeah. So with that, we have finished studying about the different types of tissues. Next, we have to learn about tissue systems. Now, what are these tissue systems? We know that in a plant, you can find different types of tissues: meristematic, permanent, simple permanent, complex permanent, all the different types we've covered. So, depending upon how these tissues are arranged within the plant, where is their location, we can classify tissue systems into three basic types. One is the epidermal tissue system. The second is ground tissue system. And the third is vascular tissue system. Since you are more familiar with tissues in animals, I will give you an example of how you can, kind of compare the plant tissues with animal tissues. If you consider tissues in animals, we have four different types. We have epithelial tissue, connective tissue, muscular tissue, and neural tissue. Similar to how our epithelial tissue is protective in function, it either lines the outside or the inside of our organs, inside of all our blood vessels and everything. Basically, epithelial tissue is protective in function. Likewise, even our epidermal tissue system in plants is primarily protective in function. Okay, so this lies on the outside. This is a superficial layer. If you can consider this to be like a stem or a root, and you take like an extremely thin slice of it and look at it under the microscope, you can see that the epidermal tissue lies outside. This is the epidermal tissue system. Okay. What about vascular tissue system? In humans, you have blood vessels that conduct oxygen, carbon dioxide, respiratory gases, nutrients, and everything across different parts of the body. Likewise, in plants, we have xylem and phloem. They are the conducting tissues. So, they are present inside. You can think of xylem and phloem like, you have that kind of pen with many different colored refills. You can click and then you can select a color, right? So, inside one common body, you have many tube-like structures. Like that, xylem and phloem will be present inside the root, stem and leaves. Okay, for convenience sake, right now, I will represent it here. Somewhere in the center, xylem and phloem will be present. That is our vascular tissue system. Everything else apart from epidermal tissue system and vascular tissue system, whatever else is present, that is our ground tissue system. It's similar to the connective tissues in animals, which connects different different organs and different tissues in humans, in animals. Similar to that, you have something called the ground tissue system. Outermost layer, epidermal tissue system. Xylem and phloem, vascular tissue system. Everything else is the ground tissue system. Now let's learn about each of these one by one, starting with epidermal tissue system. Regardless of which plant part we are looking at, root, stem, leaves or any part of the plant, the outermost covering, outermost layer is always going to be the epidermal tissue. Okay. So in the epidermal tissue, basically it is protective in function. Think about any outer layer anywhere, it offers protection. So here, this is the outermost layer. This will protect the plant body. Within the epidermal tissue, of course, you will consider epidermal cells. Epidermal cells will be a part of it. Within the epidermal tissue, you can find stomata. I'm sure you know about them. And in some cases, epidermal appendages like trichomes and root hair can be present. We learn about it in detail, starting with the epidermal cell layer. It is forming the outermost layer. It should protect the plant. It should make sure that no pathogenic organism like bacteria or fungus or virus that can cause a disease in plants gains entry inside the plant. So it is protecting it. It has to be a continuous layer without any much gaps. So the cells here are barrel shaped. If this is the outermost layer, the cells here will be like this. They are parenchymatous. They form a continuous layer like this outside. Okay, so that is epidermis. It forms a continuous layer. It has elongated cells and they are compactly arranged without much intercellular spaces. Generally, in majority of the cases, epidermis is a single layer. Like I've already mentioned, they are parenchymatous in nature. Similar to all parenchyma cells, there will be a small amount of cytoplasm, a thin cell wall, primary in nature with a large central vacuole like you see in all plant cells. Sometimes outside, depending upon which plant part it is, outside the epidermis, towards the outer side of the epidermal layer, you can see a thin layer of a waxy substance present that is called the cuticle. This cuticle prevents water loss in the form of transpiration, reduces the rate of transpiration and helps in conserving water inside the plant. Okay. Yeah. So this cuticle is present only in the aerial parts of the plant. You can find them in the stem and the root, young stems and in the leaf, not root. Cuticle is absent in roots. That's about the cuticle. Okay. So cuticle is absent in roots. We spoke about how are the epidermal cells. I told you about cuticle. Now we have to learn about trichomes and root hair. From the surface of the epidermis, if you see this is the epidermis, if you consider this to be the epidermis, okay, let's say this is the root and this is the stem. In the root, from the outermost layer, from the epidermis, you can see root hair arising. Small unicellular hair arising from the surface like this. Okay, it will be made up of a single cell. Each root hair is composed of a single cell like this. Remember when we studied about the structure of root, I told you there is a region of root hair within the region of differentiated tissue, region of differentiation. So that root hair arises from the surface itself, from the epidermal hair. The hair that you see in roots, they are unicellular. What is their function? Why should they be there? The function of the root is to absorb water and minerals from the soil. The presence of root hair increases the surface area, so water and minerals can be absorbed more efficiently. Talking about stem hair. In the stem also, you can see how from the epidermis, which is the outermost layer, in many cases you see that the stem has hair, right? On the stem, hair will arise. You call them trichomes. Now the difference between root hair and trichomes is obviously its location. Root hair is present in the root, trichomes are present in the stem. That is one thing. But stem hair, unlike root hair, are multicellular generally, and sometimes they may be branched also. Okay, so the stem hair are called trichomes. They are usually multicellular. They may be branched or unbranched. Sometimes they are soft, sometimes they are stiff, sometimes they are secretory as well. What is the function of trichomes? The function of trichomes is to help in reducing the rate of transpiration and thereby conserving water. Okay, yeah. So the function of trichomes and the function of cuticle is the same. Okay, now let's talk about the stomata. What are stomata? Stomata are openings that you can find in the aerial parts of the plant like young stems and leaves. In the stomata, when we say stomata, generally we are referring to a pore. Okay, that pore is called the stomatal aperture. Now this stomatal pore or the stomatal aperture is not always open. Sometimes they are closed, sometimes they are open. Whether the stomatal aperture is open or closed depends on two cells, that is guarded or governed by two cells, a pair of cells. They are called guard cells. I am going to outline the two guard cells in pink color here. These two are the guard cells. In dicots, you can see that the guard cells are bean shaped like I just showed you now. When you consider monocots, they are dumbbell shaped. Why do we call them guard cells? Similar to how we have security guards that check the ID card and then allow you into college and let you go out. These guard cells decide, or it is because of the guard cells that the stomatal opening opens or closes shut. Okay, so they guard that. They guard this aperture. It depends on the guard cells whether this closes or opens. Now, if you consider this guard cell, the guard cell, unlike most other epidermal cells, contain chloroplast. Okay, so within the guard cells, you can find the presence of chloroplast. In other epidermal cells, you do not find chloroplast. That is one difference between other epidermal cells and the guard cells. Also, if you look at the cell wall of guard cells, there is differential thickening. The wall of the cell is not uniformly thick. Towards the inside, it is more thick compared to the outside. This is more thick. Towards the pore, it is more thick. Now, right around the guard cell, you will have some neighboring cells that are slightly differentiated. In close association with the guard cells, you will find some neighboring cells that are called subsidiary cells. Now, whether the stomatal aperture opens or closes depends on the turgidity of guard cells. If, for example, there is sufficient amount of water available to the plant, water is well absorbed, water will move from the surrounding subsidiary cells into the guard cells. When water moves into the guard cells become swollen up. They will become turgid. When the guard cells become turgid, they will expand in such a way that this pore opens. When there is not sufficient water available for the plant, if the stomatal aperture is open, more water will be lost by transpiration. In order to prevent that, water from the guard cells will move to the surrounding subsidiary cells. When this guard cell loses water, it becomes flaccid, and when it becomes flaccid, this pore will close shut. Okay, so when the guard cell is turgid, it will be open like that because this is like a filled balloon. Imagine what happens if the balloon loses water, it will close shut. This cell will become flaccid and it will close shut. So, we have the stomatal aperture here, you have the guard cell here, and then you have the subsidiary cells. The stomatal aperture, guard cell and subsidiary cell together is what we call the stomatal apparatus. Okay, so that's what I've given here. Stomata, their structures present in the epidermis, they regulate the process of transpiration and also exchange of respiratory gases like carbon dioxide and oxygen. Each stoma is composed of two bean-shaped cells called guard cells. In the case of dicots, if it's monocots, it will be dumbbell-shaped. They enclose the stomatal pore. In grasses, they are dumbbell-shaped. Outer wall is thin, inner wall is highly thickened. Guard cells possess chloroplast. They regulate the opening and closing of stomata. I've told you all about this. right? Now, that's about the epidermal tissue system. Next, we have vascular tissue system, and then I'll tell you about ground tissue system. Vascular tissue system includes your complex permanent tissue, xylem and phloem. Depending upon how they are arranged with respect to each other, we can classify vascular bundles into two major categories. One is called radial vascular bundle, the other one is called conjoint vascular bundle. In radial vascular bundle, you have xylem and phloem present in separate radii. If you can think of this as a root and you take a section of the root and you look inside, xylem and phloem will be present. In this radius, xylem is there. Next radius, phloem is there. Here you have xylem, here you have phloem. Red color is xylem, blue color is phloem. So, in alternating radii, in different radii, you can find xylem and phloem being present. They are not present together in the same bundle. Such a type of radial arrangement of vascular bundle can be seen in roots. Conjoint is when xylem and phloem are present in the same bundle. This can be seen in the case of stems. If you take a section of a stem, vascular bundles will be present like this. Okay? Arranged inside like this. Okay, this is dicot stem. So, you're looking at one vascular bundle here. Okay, this is not dicot, this is dicot. Maybe I should draw it the other way or you'll get confused in the notes. Okay. Okay. So, what you're looking at here is one such vascular bundle. You can see how within the same bundle you have both xylem and phloem. Towards the inside, xylem will be present. Towards the outside, phloem will be present. And in between the xylem and phloem, you have a strip of meristematic tissue which we call the cambium. Remember, I've told you about cambium earlier and I said cambium is present only in dicots. So, in this conjoined type of vascular bundle, you can find both xylem and phloem within the same bundle in the same radius itself. There it was alternating. The reason we call vascular bundles open type in dicots, we call it open type because cambium is present between xylem and phloem. Because cambium contributes to secondary growth, because of the presence of cambium, this type of plant stem is open to secondary growth. It's open to becoming larger and increasing in girth. Talking about conjoint stem that is closed, you can find this in monocots. What basically happens here is each vascular bundle will have both. In monocot stems it is scattered like that. In each bundle there will be xylem and phloem present, but in between there is no cambium. That is why it is called closed. Closed because this is closed for secondary growth. In such stems you cannot find secondary growth. Think about sugarcane, maize, rice, bamboo. The stem does not increase in girth like banyan and mango tree, right? That is because they lack cambium. Without cambium, secondary growth or increase in girth cannot happen. Okay, so that's about the different types of vascular tissues. Next, we have ground tissue system. Everything other than epidermal tissue system and vascular tissue system comes under ground tissue system. In ground tissue system, you find simple permanent tissues like parenchyma, collenchyma, sclerenchyma. In different parts of the plant, you will call it with different names in different regions. When we study the anatomy of different plant organs, you will understand about it in detail. Okay, so there are a lot of names that are thrown around here. Right now, it will not make sense to you what is cortex, what is pericycle. As we understand the anatomy of roots, stem and leaves, this will make more sense to you. Okay, now let's learn about the anatomy of root, stem and leaves in that order. First, dicot root, dicot stem, dicot leaf in that order. So, I told you in the beginning that this chapter can be divided into two parts. In the first part, we learn about different types of tissues. We already finished that. This is the second part. Over here, to learn how these different tissues are organized or arranged inside the plant organs like root, stem and leaves, you have to cut them, open them up and look at it inside, right? So you will have to take sections. So we take very thin slices of the root, stem and leaves, put it on a slide, observe it under the microscope. We will observe all of these different types of tissues and how they are arranged, okay? Yeah, let's start off with the dicot root. What you're looking at here is a section of the dicot root, that is the NCERT diagram. How did we get to this section? We take a transverse section of the root. If this is a root, we cut it like an extremely thin slice horizontally like this. That is called the transverse section or the TS. When you cut it and look at it under the microscope, since most roots are, cylindrical, the section, when you cut it, the section will appear circular in outline like this. Yeah, the section will appear circular like this. Outside you will have the epidermis, inside you will have xylem phloem. What you're looking at here is one pizza-like section of that. One section of it we are learning here. Whatever is present here, we will learn. The same thing will be present over here also. So we're just learning one portion of the overall section. Alright. Now, let's start off from the outermost layer, which is the epidermis. In roots, the epidermis has an alternate name. It is also called the epiblemma. Like I've already told you, epidermis is the outermost layer in all the plant parts. Okay, root, stem, leaves, everywhere it is the outermost layer. In roots, we give it a special name. We call it epiblemma. We already know that in roots, from the epiblemma, some of the cells will give out appendages called root hair that will increase the surface area required for absorption. So many of these cells protrude in the form of unicellular root hair. Right here, the outermost layer in pink color, that is what represents your epidermis. You can also see how they are giving out these unicellular root hair. Right? Now, that's about the epidermis. What lies inside the epidermis? This portion here, everything that is represented in green color, that is called the cortex. Now, cortex is ground tissue. Okay, we learnt about epidermis, we learned about xylem phloem, except for that everything else is ground tissue. So cortex also is a part of the ground tissue system. Okay, so in the cortex you can find parenchyma cells. Epidermis generally is a single layer. Cortex, you can see how they are many layers. In the cortex, the parenchyma shape and how compactly it is arranged differs. Epidermis also is made up of parenchyma. Cortex is also made up of parenchyma. Look at how they are different in shape. Look at how they are different in how compactly they are arranged. In epidermis, you barely ever see any intercellular spaces, but within the cortex, you see a lot of intercellular spaces. Here, they are almost spherical and you do find intercellular spaces present, right? Innermost layer of the cortex, which is represented as a pink color here, is called the endodermis. Endodermis also is a part of the ground tissue system. This is the innermost layer of the cortex. Look at how it is different, how the other layers of cortex is different from this layer. Though innermost layer of the cortex or endodermis also is parenchymatous in nature, you see how here it is barrel shaped. The other layers of the cortex are almost spherical or isodiametric, but the innermost layer of the cortex or the endodermis is barrel shaped. It has cells present like this. Okay? Within the endodermis, there are no intercellular spaces, but within the other layers of the cortex, intercellular spaces was present. One very important thing that you must remember about the endodermis in roots is that in the endodermal layer, if you think about a cell, the cell will have 6 walls, right? Because it's like a cuboid. How do I show this to you? For example, let's consider this to be a cell. So you have this wall, 2, 3, 4, 5 and 6. You have 6 walls present. Imagine this is one cell. In the endodermal cells of the root, specifically in the radial and tangential walls, what are radial walls, what are tangential walls? Let's understand. How do you draw a radius to a circle? You draw a radius like this, right? So, if the cell is like this, you draw a radius like this. This and this will be the radial wall. How do you draw a tangent to a circle? You draw it like this. So, this and this will be the tangential wall. The radial walls and the tangential walls will have a substance called suberin forming a layer in the wall. Now, this suberin is impervious to water and it is waxy in nature. This suberin thickenings present in the radial and tangential walls in the endodermis of roots specifically has a special name. We call it Casparian strip. So, whenever you're asked questions about Casparian strips, you must remember that it's present in the endodermis. You must remember that it is made up of suberin. You must remember that it is found only in the root endodermis, not anywhere else. Okay, that's about the endodermis. Right inside the endodermis, what you have is called the pericycle. Okay, so pericycle can be a few layers in thickness. The pericycle also is made up of parenchyma, but unlike the cortical parenchymatous cells here, the pericycle parenchyma has relatively thicker cell wall. Okay, now there are two important points about root, especially dicot root pericycle. In dicot roots, we see taproot system. So you have the primary root. From the primary root, secondary roots or lateral roots will branch out. These lateral roots that branch out will get initiated from the pericycle. And in dicot roots, we see secondary growth happening. So in the root, cambium has to form. So the cambial ring also initiates from the pericycle. This is specific. Both of these are specific to dicot roots. Okay. One thing is lateral roots, if they have to form, they initiate from the pericycle. They are not like root hair arising superficially. They will arise from deep layer like that. And then vascular cambium has to form during secondary growth in dicots. That also gets initiated from the pericycle itself. Now talking about the vascular bundles, you can see how I've already told you earlier. Xylem and phloem are separate here, they are not present together. This is xylem, this is xylem, here you have xylem, here you have xylem. Alternate radii, you will have phloem present. What kind of arrangement do you call this? We call this radial arrangement of vascular bundles. The number of vascular bundles vary between 2 to 4. And here you can see exarch condition of xylem, which means the protoxylem faces towards the periphery, metaxylem faces towards the center. The smaller elements are facing towards the periphery and the larger ones, relatively larger ones, will be facing towards the center. That is what we refer to as exarch arrangement of xylem. Now in between xylem and phloem, okay, so look here, between xylem and phloem you have parenchymatous tissue present, right? I'll show it to you here. Between xylem and phloem there is tissue present. That tissue present between xylem and phloem is called conjunctive tissue, and that is also made up of parenchyma. And at the center of the root, you can find parenchymatous tissue that is called pith. Compared to monocot root, in dicot root the pith is comparatively smaller, so we say that it's inconspicuous, and the pith is also parenchymatous. From the epidermis to the pith we've studied all the regions. Except for xylem and phloem, we saw how everywhere it was parenchyma itself, but that parenchyma only in different regions it was modified differently. In the epidermis, it's barrel-shaped, continuous, no intercellular spaces. In the cortex, there is intercellular spaces, it's spherical. Again, in the endodermis, it's barrel-shaped with no intercellular spaces. Pericycle, there are many layers with thick walls. So, though it is parenchyma, in different regions, it's modified in different ways. Okay. Now, there is one term that you should remember, which is called the stele. S-T-E-L-E, stele. Stele is everything that includes the pericycle plus everything inside. Everything inside the pericycle. So, a stele includes the pericycle, the vascular bundles, the pith, the conjunctive tissue, everything. That's about your dicot root. Now let's talk about monocot root. There's not much difference. Of course, there's no lateral branches that arise from the pericycle. There is no secondary growth you can see. Apart from that, epiblema, cortex, endodermis, pericycle pretty much the same. There are two differences here. One difference here is with respect to the number of vascular bundles. Second difference is with respect to the size of the pith. Vascular bundles in dicot root is around 2 to 4. Here there is a minimum of 7. Okay, so there will be more than 6 vascular bundles, so you call it polyarch. In the NCERT diagram they showed 8, 1, 2, 3, 4, 5, 6, 7, 8 xylem, and obviously 8 phloem also will be present. So the number of vascular bundles are relatively more, and this pith in the center, comparatively it is more conspicuous, more developed. These are the only differences between a dicot root and a monocot root. Everything else is the same. That's about the root. Now let's learn about, just give me a minute. Yeah, dicot stem. First we learn about stem and then we learn about, dicot stem, then we learn about monocot stem. Dicot stem also is generally cylindrical. If you take a section of a dicot stem, it's going to appear circular. I want you to notice here how the vascular bundles that I had shown you earlier, over here you can make out how they all are conjoined. Within the same bundle you have xylem, phloem and cambium, and how well all of these vascular bundles are arranged in the form of a ring. This is how the section will appear. You can see a big prominent pith in the center. Now we'll take just one section of this and study about what are the different types of tissues and how they are arranged. The same thing will apply to the rest of it. Okay, we're going to just focus on this with one vascular bundle. Okay, we'll start from the outside. As always, outermost layer is the epidermis. In stems, trichomes may be present, and in some young stems you can see the presence of cuticle as well. If it is a young stem, you can see the presence of stomata as well. Okay, so these are the epidermal appendages, like the trichome is there and the stomata is there. All of these are part of the epidermal tissue system, so that you can see in the epidermis. Here the cortex is pretty complex. Within the cortex you have 3 subzones. You have a hypodermis, you have a general cortex, and you have the endodermis. Previously we saw cortex and endodermis. Now within the cortex itself you have hypodermis, general cortex, endodermis. The hypodermis is immediately present below the epidermis. I've told you earlier when I explained about 𝐂𝐬𝐂 that in dicot stems, in the hypodermal region you can find 𝐂𝐬𝐂. So here in the hypodermis there are a few layers of 𝐂𝐬𝐂 present in the hypodermis. It's represented by red color. Right below the hypodermis, you have parenchymatous general cortex. This is similar to the cortex we found in the root. Parenchymatous, rounded, thin walled with intercellular spaces. Endodermis is the innermost layer. Here, there is an interesting, important feature you should remember. It is made up of parenchyma. It is rich in starch grains. So, we call endodermis here starch sheath. In monocot root and dicot root, there is Casparian strip in endodermis. In dicot stem, you have endodermis called as starch sheath. Okay. Now, talking about the pericycle. The pericycle here is sclerenchymatous in nature. In root, we did not see 𝐂𝐬𝐂 or sclerenchyma, but in dicot stem, you find all types of tissues. There's parenchyma, 𝐂𝐬𝐂, and the hypodermis, sclerenchyma, and the pericycle, xylem, phloem, everything is there. Okay. Yeah, this pericycle is made up of sclerenchyma and it's present right on top of vascular bundles. It's present in the form of semi-lunar patches. It's given. Semi-lunar patches means half circle, semicircular patches. So you can see, I will show it to you here. Here, this is the endodermis, innermost layer. On top of that, you can see semilunar patch of pericycle. Below that, right here, you have phloem. Here you have xylem towards the inside. In between the two, here you have the cambium. Okay, so this is the vascular bundle. On top of the vascular bundle, like one topi, like one cap, this pericycle is sitting. That is why this pericycle is also called bundle cap. Okay, yeah, so that's about your pericycle. Next, let's talk about medullary rays. I showed you how the vascular bundles are arranged in the form of a ring here in dicot stem. In between the vascular bundles, whatever parenchyma is present, that is called medullary rays. This is medullary rays. In the center, what is present? This is called pith. Okay. First, let's see medullary rays. Both medullary rays and pith are made up of what is it? Parenchyma itself. Medullary rays has a few layers of radially placed parenchyma cells that is between the vascular bundles. Talking about the vascular bundles, as we already know, it's arranged in the form of a ring. It's conjoined open type. Here you have endark xylem, because if you look at the xylem vessels here, protoxylem is smaller facing the pith or the center. It is towards the inside, while the metaxylem elements that are larger faces the periphery. It depends on protoxylem. If protoxylem is facing inside, it's endark. Right now, it is endark. Okay. Talking about the pith, I showed you the pith was large, it was present in the center, and this pith has parenchyma cells with a lot of intercellular spaces. Okay, that's about the dicot stem. Now, let's move on and learn about monocot stem. In comparison with dicot stem, monocot stem is very simple in structure. You can see how there is no ring-like arrangement of vascular bundles here. All these red circles that you're looking at are vascular bundles. They're all scattered in the cortex. And you can see how there is a variation in the size of vascular bundles also. The ones that are present towards the center are larger, and the ones that are present towards the periphery are relatively smaller. Even over here, we will take just one section of this and learn it. The same thing will apply everywhere else. We will start with the outermost layer, which is the epidermis, always parenchymatous, and then here it is single layered. Thin layer of cuticle may be present. Right below the epidermis, you have hypodermis. The hypodermis here is made up of sclerenchyma. Previously in dicot stem, it was made up of collenchyma. Here it is made up of sclerenchyma. Apart from hypodermis, you do not have endodermis, pericycle, general cortex, none of that. All you have is ground tissue. Okay, within that ground tissue, the vascular bundles are scattered. And this ground tissue represented in green color is parenchymatous in nature. Okay, now let's talk about the vascular bundles. They are scattered. Okay, where is it given? It's not given. But the vascular bundles are scattered in the ground tissue. There is change in size. Peripheral ones towards the periphery, towards the outside are smaller, and the central ones are relatively larger. Here the vascular bundles are conjoined and closed. Xylem and phloem are present within the same bundle, but there is no cambium, so we say that it is closed. Surrounding the vascular bundle, surrounding the xylem and phloem, you have a sheath which we call the bundle sheath, and that bundle sheath also is made up of sclerenchyma. Sheath means covering. So if this is xylem and this here you have phloem, around that you have sclerenchyma which forms the bundle sheath. In monocot stem, within the vascular bundles you can see some water containing cavities as well. Phloem parenchyma are absent in monocots. This we already studied, right? So that's about monocot stem. If you were asked to differentiate between dicot stem and monocot stem, what would you write? In dicot stem, you have vascular bundles arranged in the form of a ring. Here it is scattered. There, the size of vascular bundles are pretty much the same in dicot stem. In monocot stem, it varies. The central ones are larger and the peripheral ones are smaller. If you have to talk about the ground tissue, over there, the ground tissue is differentiated into hypodermis, general cortex, endodermis, pericycle. In monocots, you have no such differentiation. In dicots stem, you can see that the hypodermis is made up of collenchyma. Here, the hypodermis is made up of sclerenchyma. These are some differences. If you can think of more, you can drop it in the comments. Okay, now, yeah, you can also write phloem parenchyma is present there and phloem parenchyma is absent here. Last part, we have to talk about the leaves in dicots and monocots. If this is a leaf, let's say this is like the midrib, the section that we are taking is like this. So whatever vascular bundles are there in the midrib, that will appear like this. Okay, in leaf we are not taking section like this, we are taking a section like this. Okay, so first we learn about dicot leaf, then we learn about monocot leaf. Dicot leaf is also called dorsiventral leaf because there is difference in upper surface and lower surface. because of two reasons. Okay, what is that? I'll explain. Now, in leaf, you have two outer coverings, upper surface and lower surface. So, epidermis, there are two on either sides, upper epidermis, lower epidermis. Upper epidermis has a special name, we call it adaxial epidermis. Lower epidermis is called abaxial epidermis. Adaxial, abaxial. Upper is adaxial, lower is abaxial. The way I remember it, D is for direct, B is for base. Base means bottom. So, abaxial is towards the bottom, adaxial is on top. Since leaf is an aerial part of the plant, you can see a very conspicuous thick cuticle present in leaves to reduce the rate of transpiration. The reason we call this dorsiventral leaf is because the distribution of stomata varies in the upper surface and lower surface. In the lower surface, you find more stomata. In upper surface, even if it's there, it's very little. Or in many species, upper epidermis, stomata are completely absent. They are more present towards the lower epidermis. So that creates a difference in the upper surface and lower surface. That's why it's called dorsiventral. That's one of the reasons. Okay. Now, talking about the cortex in leaf, the ground tissue in leaf is called mesophyll. Since leaf is photosynthetic organ of plant, it has to perform photosynthesis. Within the mesophyll cells, you have a lot of parenchyma that contains chloroplast involved in photosynthesis. Within the mesophyll region, you can find two different layers or two different zones. Towards the upper epidermis, the parenchyma cells shape and arrangement is different. Towards the lower epidermis, the arrangement and shape of parenchyma cells is different. Towards the adaxial epidermis, it is arranged in the very compactly close to each other with not many intercellular spaces, and the cells are also rod shaped. Cells are arranged parallel to each other. We call that palisade parenchyma. So towards the upper surface, you can see how these cells are cylindrical, rod shaped, arranged compactly parallel to each other. Towards the lower surface, you can see that the cells are somewhat spherical, loosely arranged with a lot of intercellular spaces. This is called spongy parenchyma. Similar to how there are many pores in a sponge, you have many intercellular spaces or air cavities here. This is the reason why in many dicot leaves, the color of the leaf on the upper epidermis will be more dark and lower epidermis will be relatively lighter, because here the cells are more compactly arranged. Okay, that's about the mesophyll. Talking about the vascular bundles, wherever midrib is there, vascular bundles can be found there. Here you can't say if it is, it's obviously secondary growth will not take place in leaves, so cambium will be absent. They are all present within the same bundle. Wherever midrib is there, vein is there, you can find the vascular bundles. Over here in leaves, a layer of cells called the bundle sheath will surround the vascular bundles. Usually they are parenchymatous in nature. Since in dicot leaves we see reticulate venation, there is a large midrib and its branches are relatively smaller and smaller, thinner and thinner. So if you take a section of a dicot leaf, the vascular bundle size that you see under the microscope will vary. The midrib will be the largest and its branches will be smaller and smaller. Okay, now let's talk about monocot leaf, which is also called isobilateral leaf, which means upper epidermis, lower epidermis, not much change, pretty much the same. Here the upper epidermis and lower epidermis are called the same thing. Upper is adaxial and the lower epidermis is abaxial. Here there is no difference in the distribution of stomata. Upper and lower, in both you can find nearly equal number of stomata. Here also you can find the presence of cuticle. Okay, stomata are present on both surfaces. One important thing about leaves in monocots or grasses is the presence of a special type of cell called bulliform cells. Okay, so here you can see how very close to the vascular bundles, very close to the xylem, in the adaxial epidermis, some of the epidermal cells are modified. They look different from other epidermal cells in some ways. They become larger than the other epidermal cells and colorless and empty. They are called bulliform cells. Okay, they are large, empty and colorless cells. How do these cells help? They help the plant survive when the conditions are not favorable. When there is water stress, when there is not sufficient water available for the plant, if the sunlight is more, if the conditions favor more transpiration, water will be lost by transpiration. If plant is losing all the water that it is absorbing, it will not get any water for photosynthesis, right? In order to reduce the rate of transpiration, what these cells do is they will lose water whenever there is water stress. If these bulliform cells lose water and become flaccid, since they are present in the upper epidermis, because of these bulliform cells becoming flaccid, the leaves will curl inward. If the leaves that are exposed like this curl inward, what is going to happen? The surface area exposed to the sun, exposed to the outer environment will reduce. The stomata that are open, that are exposed will also reduce. As a result, transpiration rate will come down and the plant can conserve water, right? So this will help in reducing the rate of transpiration in grass leaves. Many times questions have been asked in the NEET exam based on bulliform cells. So this is important. You should remember where they are present, how are they different from other epidermal cells, what is the function they perform, and all of that. Okay. Talking about the mesophyll, here you don't see any differentiation between palisade and spongy. All of them are similar. They are somewhat isodiametric and parenchymatous. Obviously, since they are present in the mesophyll of leaf, they will contain chloroplast, right? Vascular bundles, since this is parallel venation, you don't see any difference in size. Since in parallel venation, all the veins will be pretty much the same size, the size of the vascular bundles in the section also will appear nearly equal in size. So that's about it. What are the differences between dicot leaf and monocot leaf, dorsiventral leaf and isobilateral leaf? Firstly, epidermis, in the epidermis, stomata, distribution of stomata. In dicots, there's more in lower epidermis, less in upper epidermis, or absent in upper epidermis. Here it is almost equal. In dicot leaf, you see there is palisade and spongy parenchyma differentiation. You don't see that in monocots. In dicots, you don't have bulliform cells. In monocots, you have bulliform cells. In dicots, the size of the vascular bundles vary. In monocots, except for the midrib, it is pretty much the same. It is constant. So that is about it. As a homework, I want you to write down the difference between dicot root and dicot stem in the comments. Whatever differences between dicot root and dicot stem you can think of, I want you to write it in the comments. Okay. So with this, I will sign off from today's session. I will see you again in the next week. We will discuss another chapter, Cell the Unit of Life. And until then, take care of yourselves. Keep studying, keep smiling, stay happy. All the best. Thank you.